Can somebody confirm you can hear me?
Can hear you.
Thank you. Sorry for being a little late. Let's see. How do I share my screen here? Oh, come on. There, you should be seeing my screen now. Today is January 28th, and we should be doing Lab 5, Streaking for Isolation and Culture Media. You should also begin reading about the Unknown Project. We'll talk about the Unknown Project next week. You can get the instructions for the Unknown Project. Let's see if I can get there. I think it's week 5 of the lab. Boy, that was long. The network at Clark must be slow. So scroll down to week 5 lab and then the unknown instructions. This is the document to read. Oh, yeah, I got that there on the week 4 lab because that's where we are. All right, today we're going to be doing lab 5, module 5 on cultivation of bacteria. Any questions about what we're doing or any questions about anything in general? Where am I? There we go. All right, I'm not hearing anything, so let's begin. Can you guys see my screen? All right, I'm going to assume you guys can see my
Yeah,
screen.
we
Oh,
can.
great. Lab 5, cultivation of bacteria, culture media, inoculation, and isolation. There are only a few pages to read in the textbook before you do this lab, so please read about culture media on page 157 to 162 and obtaining a pure culture on page 162 to 163. There are some video clips in today's lab. Your learning objectives for this lab. Uh, One, be able to understand the importance of proper specimen collection and transport to the lab. Two, explain the 5 principal steps used by microbiologists to process specimens. Three, differentiate between various types of culture media. Four, be able to explain the purpose of streak plate culture technique, aka streaking for colony isolation. And then 5, define the following terms: what's selective media, differential media, and reducing media. Any questions about any of those? All right. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Oh, too far. Some microbes are sensitive to desiccation, meaning they'll dry out, and some are sensitive to the exposure of oxygen, like most anaerobes, and therefore require a special transport medium to preserve the specimen, keep it alive until it's gotten by the lab for growth. Once a specimen is received for the lab for culture, microbiologists employ the 5 principal I steps to process it. First, inoculation. 2, isolation. 3, incubation. 4, inspection. And then 5, identification. So 4, inoculation. Once they get a lab, they'll place the specimen on an artificial culture medium to initiate growth. Two, they will isolate it, spatially separating out microbial growth into distinct colonies. Incubation, placing the culture medium in the environment under conditions to facilitate its growth. And obviously, this is after you've got it um, distinct into distinct colonies and growing it out. And then four, inspection, examining the culture for the presence of microbial growth. And then five, lastly, identification, performing specific tests to ID any pathogenic microbes that have been isolated. So the five steps will be described in detail after discussing the different types of culture media. You're already familiar with general purpose media, where we talked about Na, nutrient agar, NAYE or NA, which is nutrient agar with yeast extract in it, and then TSA, tryptic soy agar. These are all rich media that allow the growth of many types of organisms, but do not allow the growth of only certain organisms, and they do not allow fastidious organisms to grow. A fastidious organism is something that requires additives into the media before the organism will grow. And so fastidious organisms don't grow on general purpose media. To grow fastidious microbes, you need to use an enriched general purpose media. For example, you can add 5% sheep's blood to TSA and then get many fastidious organisms to grow because the red blood cells require, they provide the nutrients that many, not all, but many fastidious organisms need to grow. And two media which are enriched general purpose media include blood agar plates or BAP and chocolate agar plates or CAP. Any question about any of that? So, an enriched general purpose media is both non-selective and then it's enriched, allowing fastidious organisms to potentially grow. There's also selective media, and that contains chemicals in it that inhibit the growth or halt the growth of a certain type of organism. Excuse me here. So they have something in it that stops the growth of certain class of organisms. Keep in mind when two things when you're using selective media. First, selective media isn't necessarily perfectly selective. If given enough time, organisms that are selected against may grow on the dish. And second, even those organisms that are supposed to grow in the selective media, they may grow smaller colonies or grow more slowly than they would on a general purpose medium. Any question about any of that? All right. There's also differential media, and this contains chemicals that produce visible differences among different groups of organisms when they're grown on that medium. These differences are unrelated to how well the organisms can grow on the medium. But when you look at the plate, you might see two different colors from two different organisms growing on the plate. The differential culture media allows dissimilar bacteria to be visually distinguished from each other. And lastly, there's reducing media, which I guess isn't quite the last media we'll talk about, but the last media for today. Reducing media promotes the growth of anaerobic bacteria because low or no oxygen conditions are required to grow these organisms. So reducing media has something in it that removes oxygen. It's important to note that a culture media can be classified into more than one category. For example, McConkey's agar is both selective and differential. And then blood agar is both an enriched general purpose media and a differential media. Any question about that? So here we're seeing McConkey's agar. If the organism grows on McConkey's agar, it's likely to be gram negative because McConkey's agar selects for gram negatives. It has chemicals or additives to it that prevent the growth of gram positives. And then if the organism grows on the McConkey's agar, it can look different, like this growth looks pinkish, and that growth looks white or off-white. And that's because it's differential for um, lactose fermentation. If the organism can ferment the sugar lactose, it'll lower the pH and become acidic, and that will change the color to pink. Both the growth will be pink, and then the plate around the growth will also be pink. And then if the organism cannot ferment the sugar lactose, it'll be its normal color, which in this case would be white or off-white. Now, because it's growing on McConkey's, it is not fermenting the sugar lactose here, but remember it's probably gram negative because it is growing on the plate. So McConkey's is both selective and differential. Blood agar is a media which is an enriched general purpose media because it can grow a fastidious organism like Streptococcus pyogenes, which requires the additives that you can find in the blood before Streptococcus pyogenes will grow. It can grow other organisms, like one that Staphylococcus epidermidis, maybe, which does not require the blood, but it will grow in the blood agar plate. And then it's differential by its hemolytic pattern that can be observed on the plate. You can have beta hemolysis, and that's complete hemolysis, and that is where the red blood cells are completely lysed. And you see this clear region on the plate, either in between the beta. The beta is the growth of bacteria. The clear region is the beta hemolysis. And if you hold this plate up to something, you can actually see through the clear region. I usually hold it up to a clock or to the light and then can see the light or the clock behind the clear region. There's also alpha hemolysis, and that's the partial hemolysis of the blood. So here we have the red blood, and let me blow this up a little bit. And as you get really close to the alpha, which is the non-white streak of bacteria growing, you can see a darkened ring around the alpha. The darkened ring is really visible inside the alpha because it's entirely dark. That darkening, or sometimes it looks greenish, here it looks dark, is from the partial hemolysis of the red blood cells. And as you can notice, around the beta hemolysis, we have a darkening, and it's normal to see a ring of alpha hemolysis around beta hemolysis. Let me shrink that down now. And then we have the third type of hemolysis called gamma hemolysis, and there's no hemolytic activity of these cells, meaning the red blood cells are red up until the growth of the bacteria. No hemolysis or gamma hemolysis. Any questions about hemolytic activity and blood agar plates? All right. So this table is showing you the different types of media. We have general purpose media, including tryptic soy agar or TSA, nutrient agar, NA, nutrient agar with yeast extract or NA. And then we have differential and enriched medium, and blood agar plate is one of those. It's differential because it can differentiate on hemolytic activity. And it's an enriched plate because it has blood in it, which supplies the nutrients for many fastidious organisms to grow. There's also a general purpose, excuse me, an enriched general purpose media, and chocolate agar or cap is that way. It supplies the blood, but the blood are lysed, making the plate look brown and it kind of looks like chocolate, why it's called chocolate agar. And so the blood cells can supply the nutrients which are required by some fastidious organisms to grow. And so that's why chocolate agar is an enriched general purpose medium. It is not differential. Okay. All right. And then there's McConkey's EMB and HE, McConkey's MAC, EMB eosine methylene blue, and HE hectone agar. They include chemicals or additives in the media to make them selective and differential. All three of these plates are selective for gram-negative bacteria, meaning they will grow on these plates. Gram-positives tend not to grow on these plates. All three are also differential in that they have a chemical in it, allowing you to see if lactose fermentation occurs. If lactose fermentation occurs on McConkey's, it'll turn pink. If lactose fermentation occurs on EMB, it'll be either a metallic green color or a darkening of the plate, usually blackish, but a darkening. And on hectone enteric agar, it if it is differential for lactose fermentation, it will look salmon colored. Salmon is a yellow orange color. And if the bacteria cannot ferment the sugar lactose, they will be their normal color, and the growth around the plate will be normal. Like McConkey's, well, you'll see pink growth, but the plate color around it will also be pink. And the same with EMB, it'll be either metallic greenish or a darkening, both of the growth, meaning the colonies, and then the growth around the plate. And the same with hectone enteric agar, except it'll be salmon colored. Any question about any of that? And then we have media which is only selective. PEA or phenyl ethyl alcohol is a selective media. This is selective for gram positive, meaning gram positives can grow on PEA, but the phenyl ethyl alcohol gets in the cell wall of a gram negative and then prevents the cells from growing. So this is selective against gram negative, selective for gram positives. And then there's mannitol salt agar. This is a selective and differential media. It is selective because it selects for high salt. This an organism, if it can grow in high salt, tends to grow in mannitol salt agar because it has 7% sodium chloride in it. It is also differential because you can differentiate the growth on the plate into those that can ferment the sugar mannitol. They will turn yellowish around the plate, meaning a yellow halo around the growth or around the colony. And then those that cannot ferment the sugar mannitol, they will be the normal color of the plate, which is pinkish. Uh, it says it used to distinguish Staphylococcus aureus from other Staphylococcus species. That is true, but do realize that other bacteria can grow on mannitol salt agar besides Staphylococcus, like Micrococcus luteus can grow on a mannitol salt agar plate, and it is not of the Staphylococcus family. All right, any questions about the different types of media? All right, here we've inoculated a plate with some growth and then incubated it, and you can see the growth on the plate. This is just microbes from the air. Isolation is important Louis Pasteur developed the concept of a pure culture. His ability to get a pure culture was actually quite difficult. And what he did was he took a liquid growth of culture and then diluted it, replica plated it 10 times, diluted that one more time, replica plated that dilution 10 times, diluted that dilution another tenfold, and then made 10 replica plates of that. And so what he did was he made a series of different dilutions and then replicated each of those dilutions 10 times. And if he got growth in only one tube and 9 of the other tubes did not have any growth, he correctly concluded that the one tube of growth was likely to be a pure culture started by one or a few cells. And he was correct that if you do that and you get a dilution where only one tube has growth and 9 of the other tubes do not have growth, he likely got a pure culture. Uh, Dr. Koch developed the streak plate method where he used a solid plate, solid media, and then streaked out for colonies. And if he took from a pure ice, well, not a pure, but a separated colony, meaning no other colonies were touching it, and then he streaked that out on a second plate for isolated colonies, and then he took the cells that went through two colony isolation events, you're likely to get a pure culture. And it's likely to be over 95% chance that it's a pure culture, meaning one of the colonies was likely formed from one cell or very few cells, which are derived from one cell. And that's why you do it 2 times. And that's because if you only did it 1 time, about 30% of the colonies will be formed from more than one cell. But if you do it from two isolated colonies, meaning streak it out twice and get isolated colonies, and you take cells from one colony from the second plate, then you will likely have a pure culture, greater than 95% chance. And that's such a good way of getting a pure culture, we still use it today. So what you do to get a pure culture is you take your mixed growth, meaning mixed cells, and then streak it out for colony isolation on sector 1, and then sterilize the loop, pass it through a flame, or put it into a Bacto incinerator, get it red hot, cool the loop, pick up the growth from the first streak. What you do is you start in quadrant 2, come back to quadrant 1, pick up some growth, streak it out on quadrant 2. And this isn't quite done correctly because what you should do when you're streaking it out in quadrant 2 is you start in quadrant 2 to cool the loop and then come into quadrant 1, but you only come into quadrant 1 where the cells are dilute. So as you're streaking quadrant 1 here, the cells will be more dilute in this section of the quadrant, and they'll be more concentrated in this section of the quadrant. You shouldn't go into the high dilution where the cells are more concentrated in quadrant 1. You should only go where the cells are lower in quadrant 1. So you should really only go in this region here. And you should do it 3 or 4 times. This is doing it 1, 2, 3, 4, 5 times, a little more than you should. 3 or 4 times. Go into quadrant 2 and just only come this far. Don't go to this section. Streak out quadrant 2, and it's only showing you 1, 2, 3, 4, 5 streaks. You should streak it out about 20 to 30 times in each quadrant. and then sterilize your loop. Start in quadrant 3 and then go into quadrant 2. But once again, don't go into the high density region of quadrant 2. Go into the lower density region of quadrant 2 about 3 or 4 times. So come in this region, come here, and then streak it out in quadrant 3. Quadrant 4, they've actually done correctly. They streaked it out in quadrant 4, went into quadrant 3 where it's on low density in quadrant 3. Go about 3 or 4 times. They did a little more than they should. And then streak it out 20 to 30 times in quadrant 4. The idea of this different quadrant system and doing the streaks is someplace along the line, either quadrant 1, quadrant 2, quadrant 3, or quadrant 4, you will dilute the bacteria so that in one of the quadrants you'll be streaking and you'll streak down individual isolated cells, which will give rise to individual isolated colonies. In this case, it happened in quadrant 4, but it doesn't matter where it happens. If the solution you initially used had been a little bit more dilute, it wouldn't have happened in quadrant 4, it would have happened in quadrant 3. And if it had been even more dilute, it would have happened in quadrant 2. And if it had started out very dilute, you would have gotten isolated colonies in quadrant 1. Any question about any of that? Now remember, to get a pure culture, you take from an isolated colony in this plate and streak it out again on a second plate to get isolated colonies. And when you get a second plate having isolated colonies, you take cells from one colony to set up your stock culture. And then 95% of the time or higher, you will have a pure culture. And yes, you can't ever quite say with 100% confidence you have a pure culture, but it'll be over 95% of the time. And that's good enough in microbiology. If you were to take a colony from only one plate, meaning you streaked it out only one time to get isolated colonies, your chance of getting a pure culture is only about 70% of the time. And that is not good enough in microbiology. Any question about any of that? All right. The next I procedure is incubation. Recall from lab 1 that a culture media contains chemical nutrients necessary for growth of the microbe in vitro, but you must additionally provide appropriate physical environment for the microbes to grow. You can have a complex incubator like this one shown here, which can actually, these dials can control how much oxygen will be in the incubator for the cells to be exposed to, as well as how much CO2 is in the incubator for the cells to be exposed to. In our microbiology lab at Clark College, we don't have any fancy incubators, so our incubators only control the temperature, which this one does. It does control the humidity, meaning we see this pan there. You put water in that pan, that will allow the water to evaporate, increasing the humidity in the incubator. That's important because if you did not put in the pan of water, the plates would dry out after only a few days of growth. But if you have the pan of water in there, the plate will last at least a week. Sometimes the plate can last about as far as 2 weeks before it'll dry out. Oh, and then the other thing is the incubator allows you how much time to grow it for. And what you do is you just come in and you allow it to grow for the time of length you want and then pull it out. Usually in the lab, because we have the lab on Tuesday and Thursdays, most students grow it for 48 hours, but you can grow it for like a day later. You'd have to come into a lab, like you streak it out on Tuesday, you'd have to come into the lab on Wednesday and just ask the instructor in the lab if you can come in and take a look at your growth and then pull it from the incubator. And as long as he says yes, you can come into the lab and do that. All right. Then there's the I step of inspection. There are obvious visible colony morphological variations between different species of bacteria growing on the plate. Microbiologists can describe the appearance of the colonies by a variety of descriptions. The size of the colony, the shape, the color, the margin, the elevation. Let's come up to this plate here. And you can see different colony characteristics. This is obviously a different colony than that one, as well as different than that one and different than that one. You cannot identify the species just yet, though, because to identify the species, you have to run further tests. Identification. Once the relevant microbes have been isolated in a pure culture, further studies are required, such as obviously staining, like gram staining, and then running further tests to determine the species that is growing. Any question about that? And that's called identification. All right, know the terms of this lab? In this lab, you're not going to be doing any of these procedures because everything we're doing in this class is online. But we have a little video for you to watch on the inoculation of an agar plate using the streak plate culture technique. You only need to watch this video to 4 minutes and 15 seconds. You should note that this person only used 3 sectors to make the streak. We showed you 4 sectors. Doesn't matter. Both of them have streaked out the plate for colony isolation and then obtain isolated colonies. We have a little video on beta hemolysis, so watch that. A little video, very short, on alpha hemolysis, watch that. And a little video on gamma hemolysis, watch that. And then we have a little video discussing McConkey's agar. So watch that. You only need to watch it into 3 minutes and 50 seconds of the video. And then we have a little video discussing mannitol salt agar. So watch that video. We no longer have the virtual lab on practice preparing a streak plate because it required the use of Flash, and Flash is no longer running. It's no longer supported by Adobe. And so we do not have a virtual lab for this lab. All right, I think I've pulled out the questions for you. So let me go to the worksheet. I've changed some of the questions too. So the worksheet for Lab 5, filling your answer to the worksheet for Lab 5 by this Saturday, 11:59 p.m. And by the way, I should mention that one of the first lab to last week, which I guess would be Lab 3, I have got it graded, and so it will be due. If you did not get 100%, you may turn it in by this coming Saturday at 11:59 p.m. for full credit. Lab 4, I'm still working on. If I get it done by Wednesday morning, it too will be due this Saturday. If it takes me longer to grade it than Wednesday morning, I will not make it this Saturday that it's due. I'll make it the next Saturday that it's due, the second grading of the lab. All right. So question 1, describe a natural circumstance where Staphylococcus species of bacteria might benefit from tolerating salt. And do not say something like growing it on mannitol salt agar because that is not a natural circumstance. 2, how might hemolysis benefit a hemolytic organism? Remember, hemolysis is the lysing of red blood cells. How may that benefit the bacteria causing the hemolysis? And then 3, Imagine you have a culture containing a single bacterial species. This culture may be one of several possible species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to identify the unknown bacterial species. That is, you need to name two properties from each of these plates to help you identify the species. 4. What is the main goal of streaking for colony isolation? 5. What happens to the growth on the plate if you do everything correctly except you forget to sterilize the loop in between sectors when streaking for colony isolation? You need to be specific discussing the growth on each sector of the plate. And note that it says you forgot to streak in between the sectors. That means you did, not streak, meaning you forgot to sterilize the loop in between sectors, but you did sterilize the loop before sector 1, because that's not between sectors. Any question about that? And it does say everything was done correctly. So you did streak it, not streak it, you did sterilize it before sector 1. And another thing is obviously you did it after the very end too. That's not terribly important. But just tell me what the growth on the plate will look like in each sector. And you have to be specific. 6. What happens to the growth on the plate if you do everything correctly, but you transfer from the stock culture to inoculate each of the sectors? If you did it correctly, you'd only transfer from the stock culture on the very first sector. So that's what's done wrong in here. Be specific once again, discussing the growth on each sector. 7. Suppose you successfully streak a plate and obtain isolated colonies of two different bacterial species. That is, you are streaking a culture of mixed cultures. How can you use this plate to create a near pure culture of only one of the bacteria? You must be specific in your answer. For full credit, you need to have a large enough discussion that I can make out what you're doing and how you obtain deep pure culture. 8. In the table below are a variety of agar plate media available to microbiologists to cultivate bacteria. To note whether each medium is non-selective, a general growth media, selective media, and or differential media by placing an X in the appropriate column or columns. You may use the table of media in the lab module to answer these questions. So nutrient agar, is it non-selective, selective, or differential, or potentially all three? I'll tell you off the top of your head, you'll never get all three because you can't be both non-selective and selective. And then tell me for tryptic soy agar, tell me for chocolate agar, tell me for blood agar plates, that's TSA with 5% sheep's blood or BAP. Tell me for McConkey's agar, EMB agar, HE agar, MSA agar, and then PEA, phenyl ethyl alcohol agar. Any questions about question number 8? And then for 9, tell me which agar plate or plates were inoculated without using a streak plate method, meaning you did not streak for colony isolation. Plate A, plate B, plate C, or any combination of those 3. Any questions about the worksheet? If not, I'll be here until 4 o'clock to help you with any questions you might have, or until the last student logs out. So if you have a question, make sure you ask before the last student logs out. Any questions about this lab? All right, get started in the lab.
I wanted to ask a question about number 7
Uh-huh.
on the lab. So you mentioned that you want us to be specific on what we're doing and how we obtained the culture itself, but what, I just want to make sure that I'm completely understanding the question. What else do I need to include?
Well, I'm not sure what you don't understand, so I'm not entirely certain what you're
Okay.
doing. You have to list all the steps you do to get a pure culture.
Mhm. Okay. And you just want us to list it out step by step, essentially.
You can do it in a sentence format, but or a paragraph
Okay.
format, but you can list it out step by step also. Like step one, you do this. Step two, you do that. Either
Okay.
way.
Okay. Thank you.
All right.